Yeah. Sorry, it got interrupted. So I was talking about basically, I started by telling what was covered during the week, which was biomolecules, central dogma, regulation, and non coding RNAs. And then I discussed how some elements are important for mammals. Moving on to what elements form an important part in biomolecules. So, as I said, hydrogen, carbon, nitrogen, oxygen, and phosphorus. They mostly form the backbone of major biomolecules, for example, nucleic acids or like polysaccharides and lipids, things like that. Sodium, magnesium, potassium, calcium, and chlorine are some of the organic ions that play an important role in osmosis and in things like neuronal transmission and information of action potentials. And then we talk about manganese, iron, copper, zinc, cobalt, which are some metals which form an important centers in the enzymes which function in redox reactions in the human body or in general in living organisms. Coming to water, as you might already be aware from your high school, that water is a polar molecule, and that is because oxygen, which is connected to these two hydrogens to form the water molecule, is more electronegative than these hydrogen atoms. And so the shared electron pair between hydrogen and oxygen is actually pulled towards the oxygen atom, which makes oxygen slightly more negative than hydrogen, and hydrogen is slightly more positively charged. And this is what leads to the formation of hydrogen bonding as well, because this slightly negatively charged oxygen can then form these weak bonds with the slightly positively charged hydrogen, which are called hydrogen bonds. And they can also form these bonds with other things, for example, dissolved salts such as NaCl, in which the sodium is, which is the positive ion, is connected to the oxygen, and chlorine to these hydrogens. Moving on to dehydration versus hydrolysis. So, dehydration is a type of, as the word suggests, it's a type of reaction in which water molecule is removed, and it basically leads to synthesis of bigger molecules. For example, you have two molecules here, and in which if you remove the hydrogen and the OH. So, if you remove the water molecule, you get this bigger molecule, which is comprised of these two smaller ones. And the reverse of this is basically hydrolysis. If you add water to a bigger molecule, then it can become two small molecules. So, breakdown of bigger molecules by the addition of a water molecule is called hydrolysis. Then, coming to biomolecules. So, in this week, they talked about four major biomolecule types, including nucleic acids, proteins, polysaccharides, and lipids. So, when you talk about biomolecules, you also talk about how they are made. So, for polysaccharides, they're mostly built up from simple sugars. For example, in the left hand side, you can see these simple sugars like simple carbohydrates, which are called monosaccharides or disaccharides, fructose, galactose, glucose, and they give rise to these complex carbohydrates, which are called polysaccharides. For example, fructose, when multiple fructose molecules come together, they form glycogen. Galactose forms starch or amylose, and glucose forms starch and amylopectin. Fatty acids come together to form fats or lipids. Amino acids are building blocks for proteins, and similarly, nucleotides are building blocks for nucleic acids like DNA and RNA. So, coming to DNA, which is the deoxyribonucleic acid, it is formed, its backbone is composed of a pentose sugar, which in the case of DNA is deoxyribose, but in the case of RNA, it's ribose. And it's form of a pentose sugar, which on its first carbon, it has a nitrogenous base attached to it, and on the fifth carbon, it has a or on the fourth carbon, it has a phosphate group attached to it. Okay. So these nitrogenous bases are these four bases. They could be adenine, thymine, guanine, or cytosine. And as you might already be aware, adenine and thymine form a pair in the DNA, and guanine and cytosine form another pair. So ATEs forms a pair and GC forms a pair. And adenine and thymine are connected by two hydrogen bonds, as you can see in this figure, and guanine and cytosine are connected by three hydrogen bonds. So this is what a DNA molecule looks like. There are two strands which are helically coiled around each other to form a double helix. And here you can see A is connected to T and G is connected to C. And in yellow is the phosphate backbone. So Watson and Crick model of DNA looks something like this. So this is the phosphate backbone that we talked about. This is the phosphate molecule, and here is the pentose sugar, and here is the nitrogenous base. So to the first carbon is connected the nitrogenous base, and to the fifth carbon is connected the phosphate group. So here, the DNA molecule, or the strands of the DNA molecule, they have directionality, as you might already be aware about this also. So they run from the 5' end to the 3' end. So in the 5' end, it's the side where it ends at the phosphate molecule because it's connected to the fifth carbon. And at the 3' end, it's the one where you have this free O group, which can, where another phosphate group can attach to this O here. Like this one is attached here, another phosphate group can come and attach and can continue the strand. So from 5' end to 3' end is the directionality of the uh, and the two strands have opposite directionality. If this one is from top to bottom 5 to 3, then this one is from bottom to top 5' end to 3' end. Moving on. Oh, also to mention here in this figure, you can see here that this GC pair is kind of bonded to the AT pair as well. And this kind of bonding is called pi-pi bonding, in which the electron cloud present in between these two base pairs is actually shared by the two base pairs. So that also improves or provides more stability to the structure of the DNA molecule. Okay. So comparing the DNA with the RNA, so in the DNA molecule, you have these four types of base pairs, or the nitrogen uh, bases: cytosine, guanine, adenine, and thymine, CGAT. And in RNA, you do have cytosine, guanine, and adenine, but in place of thymine, you have uracil in RNA. And as you can very clearly see from the image, DNA is a double helix. It has two strands coiled around each other, but RNA is a single stranded helical structure. It only has one single phosphate and sugar backbone. So you might think from this, or it's kind of obvious, that RNA is a little more unstable and a little more reactive as compared to the DNA, because here all these nitrogenous bases are exposed, but in DNA these nitrogenous bases are not exposed, they're actually inside the helical structure. Moving on to the next biomolecule, which is protein. Protein is actually made up of amino acids, and this is what an amino acid looks like. So there is carbon in the center. On one side, it has a carboxyl group. On another side, it has an amine group, which is NH2, and there is a variable group R present here. So this variable group can be joined by different carbon chains, and it gives rise to different amino acids. In humans or in living organisms, we have 20 different amino acids, and they can be categorized as non-polar, polar, basic, or acidic depending on their tendencies. So non-polar amino acids basically will be the ones that will be not soluble in water because they're non-polar, and the polar ones will be soluble in water. And so this is about proteins and their building blocks, which are amino acids. So proteins can have different structures. If you're talking about a primary structure of the protein, it will be a polypeptide chain, which will look somewhat like a straight chain. But when this chain gets longer, it can assume structures like secondary structures, like a alpha helix. It can either form a helical structure like it's found in the RNA, and it can form this helix, or it can either form beta sheets in which two different polypeptide chains can be connected together in parallelly to form a sheet like structure. And now these different secondary structures can come together to form a tertiary structure. For example, it's shown here. And in this tertiary structure, both alpha helices and beta sheets are present to form this tertiary structure. And multiple tertiary structures can form subunits of a bigger protein and form a quaternary structure. And this structure of the protein is very important because it decides where the active site, since a lot of enzymes are actually proteins, it decides where the active site of the protein is and what shape it takes. So it depends on which molecules it can react with. Coming to lipids, as you might already be aware, lipids are actually made up of fatty acids, as I already mentioned, and they mostly form cell membranes. They form these bilayer sheets in which there are these polar heads which are attached to these hydrophobic tails. And these polar heads can actually interact with water, but these hydrophobic tails, they come to face each other because they cannot interact with water. So this is how the bilayer is formed, because these hydrophobic tails, they face each other, and the polar heads face away from each other. And this, you can see, is how a protein or a lipid bilayer forms the covering of a cell, or forms the cell membrane. And you can see in this figure that these tails are formed of these fatty acids, which are actually not soluble in water, so they are the hydrophobic part. And here you have a phosphate group, and you have a glycerol molecule in the middle. So this glycerol head, which is all, which it's hydrophilic, and these tails, the fatty acid tails, are actually hydrophobic. Lipid membranes, they also sometimes have protein channels through them, which allow the flow of substances in and out of the cell. So these can be ion channels, or channels for other molecules. So they allow the movement of ions in and out of the cell, which is very important for many processes, such as the generation of action potentials, in which you need the movement of sodium and potassium ions in and out of the cell based on their um, against their potential, or along their osmotic potential. Moving on to the last type of biomolecules that we'll be discussing, which are actually polysaccharides. Polysaccharides, as I already mentioned, are made up of monosaccharides. So, as you can see in this image, monosaccharide is a single sugar molecule, and when two of these connect, they form a disaccharide. And polysaccharide is basically made up of multiple sugar molecules. And here we have an example where glucose molecules come together to make up starch or cellulose. It can make up both of these molecules. It just depends on how these molecules pair together. Is If they pair in one way, then they form starch molecule. If they pair in another way, they can form cellulose. So one of them is digestible by humans, and one of them is non-digestible, or it's undigestible by humans. It cannot be digested by us. Okay, moving on to more complex molecules. So, as we discussed polysaccharides, now there are also mucopolysaccharides, which are also called GAGs, or glycosamine glycans. For example, heparin sulfate, dermatan sulfate. These are basically complex sugars that are produced by cells, and they are mostly transported to the extracellular space, and they can be found mostly on the surface of these cells. For example, you see epithelial cells here. So the basement membrane here is filled with like chondroitin sulfate, keratin sulfate, heparin sulfate, these different mucopolysaccharides, which provide stability to the structure. And other things are like these aggregants. They're made up of proteins and sugars. The backbone is made up of these uh, these sugars, and these proteins, they hang out from that. So these are called aggregants. Okay, yeah, and they give like a bottle brush appearance. Coming to an important uh, concept that this was discussed during this week is central dogma. Central dogma is basically talks about how the genetic instructions, or the genes that are present in the form of the DNA sequence, they are transcribed into mRNA, and then they are translated into a protein. So this process is referred to as central dogma, and you can look here. So from the DNA comes the mRNA, which is also called the messenger RNA, by the process called transcription. And then by translation, the mRNA is converted into the peptide chain, or the polypeptide, or the protein. Now, this process in which DNA forms RNA is called transcription, in which you are transcribing information in the same language, basically. So, in the language of the four nucleotide bases. But from RNA to protein, it's called translation, because the language changes. From the language of these codons, or these nucleotide bases, you reach the language of amino acids. So, that is translation. So this is another way of showing central dogma. DNA via transcription gives rise to RNA, which by translation forms protein. RNA can actually sometimes in these retroviruses can give rise to by reverse transcription can give rise to DNA. DNA can give rise to more DNA by replication, and RNA can also is of different types like mRNA, rRNA, and tRNA, which we'll be discussing further in this slides. You can see here, this is a eukaryotic cell. In a eukaryotic cell, transcription, which is the formation of mRNA from DNA, happens inside the nucleus, but translation happens outside the nucleus in the cytoplasm. Okay, so a question that might come to your brain, that if you're actually using DNA to in the end form proteins, why don't you use DNA directly? Why do you have this RNA in the middle? Why do you have uh, this one level in the middle in which you're transcribing DNA to RNA and then translating it to protein? Why can't you directly make protein? So that is because RNA actually serves as a control, and it helps you in getting either huge amounts of proteins or into getting very little amounts of protein as is required by the cell. And the other important part is that DNA should be kept safe, because DNA is the master copy which contains all of the genetic instructions. So it is very important for the cell or for a eukaryotic cell to keep this DNA safe and to not expose it to any external factors. And that is why DNA should be safely kept inside the nucleus, where it carries all the instructions. But it can be opened and made into smaller copies of RNA, which can then be translated so that the master copy is safe. So, how is transcription different in eukaryotic cell versus in prokaryotic cell? So, here is an example of a eukaryotic cell, and here is a bacterium. In a bacterium, as you might already be aware, that there is no nucleus. DNA is just present as a double helix in the cytoplasm itself. So, one important part is that the transcription and the translation, they can happen simultaneously, and they are not separated in space or in time. So here you can see in this image that the DNA is being transcribed into RNA, and the RNA is simultaneously being translated into a polypeptide chain. So these two things can happen simultaneously. But in eukaryotes, since there is a nucleus and transcription happens only inside the nucleus, then transcription is actually spatially different from translation, because translation can only happen after transcription is complete. Another thing is that in prokaryotes, mRNA, once it's formed from the DNA, does not really undergo any form of processing, because DNA in bacteria or in prokaryotes does not have introns and exons. But in eukaryotes, as you can see in these two different shades of purple, in eukaryotes, the DNA actually has two different types of sequences. One are called introns and one are called exons. And in the final mRNA, you actually just need exons. So this mRNA needs to be processed before it actually forms the protein or the polypeptide chain. Now, to discuss deeply about introns and exons, introns are mostly those sequences which do not code for protein. They might code for other RNAs, but they do not code for any polypeptide chain or any proteins. But exons are the part of the sequences that actually code for proteins. So, as I discussed, how are eukaryotes different from prokaryotes in transcription? In eukaryotes, the DNA has introns and exons, as I already mentioned, but DNA does not have introns in prokaryotes. All of it codes for proteins. Transcription and translation, they happen in the nucleus and in cytoplasm, respectively. Transcription happens in nucleus, translation happens in cytoplasm. But here, the cell is not compartmentalized in prokaryotes, and so transcription and translation can happen at the same time in the same space. There are three types of RNA polymerases in the eukaryotes, but only one type of RNA polymerase in prokaryotes. I will come later to what is an RNA polymerase. It's actually a very important protein which leads to the formation of mRNA from DNA. And gene regulation is complex in eukaryotes, and it can happen at different levels, such as splicing or chromatin modifications. And gene regulations in prokaryotes is pretty basic, and it only happens at the level of transcription initiation. Okay. So moving on to what are the different players in transcription. So we've been talking about transcription. So what are the different things that play a role in transcription? So one, first of all, DNA is required, which is the as I said, it's the genetic instruction. From DNA only you can form mRNA, right? So DNA is required, and DNA consists of these different parts. At least in eukaryotes, there is a promoter sequence. There is the gene, the actual gene which needs to be transcribed, and then there is the terminator sequence. So promoter sequence is actually where the RNA polymerase comes and binds, which is the protein I was talking about. And RNA polymerase leads to opening of the DNA helix, and then it actually leads to the formation of the mRNA. It leads to the joining of the nucleotide bases and forming the mRNA over this template strand of the DNA. And then terminator is where the transcription stops, or where the DNA comes to an end, or the gene comes to an end. Pre-mRNA in eukaryotes is actually the mRNA which still needs to be processed. It has both exons and introns. You can see here there are these exon portions which actually code for polypeptides, and introns which are not required. So this is a pre-mRNA which has both exons and introns, and after splicing, it forms this mRNA, which is good to go to form the polypeptide chain. And spliceosome is actually the structure which leads to this splicing and cutting out of introns and connecting of the exons together. Coming to transcription in more detail, so as I mentioned, this RNA polymerase comes and binds to the DNA strand, and it leads to opening up of the DNA helix, and and it moves along the helix and keeps joining the nucleotide bases, forming the RNA. To keep in mind, the RNA polymerase always forms RNA from 5' end to 3' end. Therefore, the template strand that is used of the DNA is the one running from 3' end to 5' end. Then the coding strand, or the strand which is similar to the RNA being formed, which is it's called the coding strand, it runs from 5' end to 3' end. So the only thing to keep in mind is that the RNA polymerase forms mRNA from 5' to 3'. So here you can see the coding strand is actually similar to the RNA strand. It's just that the T's are replaced by the uracils in RNA. Okay. So there are these 4 steps for transcription. Number 1 is preparation, where the RNA polymerase comes and binds to the DNA, forming the transcription bubble. Then the mRNA is started to be made. So basically, it opens up the helix and it starts attaching the nucleotide bases, forming the mRNA. Then, after termination, the mRNA detaches and the RNA polymerase also detaches from the DNA, and this mRNA is now formed. And this mRNA then gets modified, as I mentioned, by the spliceosomes to cut out the exon portions, and to join the exon portions and cut out the introns. So, the spliceosome helps in that in cutting out all of the introns. And then at the a 7-methyl guanosine is attached at the 5' end, which is the 5' end cap, and at the 3' end a polyadenine tail is attached. So a lot of adenine groups are attached at the 3' end. This is to protect the RNA from any kind of reactions that might happen when it exits the nucleus into the cytoplasm, because the cytoplasm is a very active area with a lot of enzymes moving around in that. So that's why you require this methyl cap at the 5' end and polyadenylation at the 3' end. So I've attached this short video that explains transcription in much more detail so that it becomes more clear to you. Do let me know if you can.
Transcription takes place in 2 broad steps.
Audible to you.
First, the enzyme RNA polymerase forms a pre-messenger RNA molecule. This happens in the nucleus by using base pairs complementary to the base sequence of the DNA template strand. In the second part, the pre-mRNA is edited by more enzymes to produce a desired mRNA, which is the final product of the transcription. So let's look at the first section of this process, which is the formation of the pre-messenger RNA, which essentially takes place in 3 steps: initiation, elongation, and termination. Now, the main enzyme that is involved in this process of transcription is the RNA polymerase. The RNA polymerase has the main job of copying the information of the DNA to a new RNA molecule. RNA polymerase builds the new strand from the 5' to the 3' direction, adding new nucleotides to the 3' end. So the first step in the formation of a pre-messenger RNA molecule is the initiation, in which the enzyme RNA polymerase binds to the promoter region of the DNA. Now let's suppose this is a gene that is shown in the green that needs transcription. At the beginning of each gene, there is a sequence of base pairs known as the promoter region of the gene. The promoter region serves as a guide which tells the enzyme RNA polymerase where to attach. Once the RNA polymerase binds to the promoter region, it separates these 2 strands of DNA, providing the single-stranded template needed for transcription. Only one of these strands is used in the process of transcription, which is known as a template strand. This strand always runs from the 3' to the 5' direction. This is because of the fact that we just discussed. That is, that RNA polymerase can only synthesize the new strand from the 5' to 3' direction. So it can only use a strand of the DNA running from the 3' to 5' direction as a template, as the new strand that is copied is always copied in the anti-parallel direction. So after the RNA polymerase binds to the promoter region of the DNA, the second step in this process is elongation, which is the process of copying the bases on the DNA template strand to form the new complementary RNA strand. The RNA polymerase uses nucleotides according to the base pair complementary rules to create a RNA copy. In the previous videos, we learned how the DNA is copied by DNA polymerase to form an identical new strand of DNA. In that case, the DNA polymerase added the base thymine when the template strand had the base adenine. and it added the base cytosine when the template strand showed the base guanine and vice versa. But in case of transcription, the RNA molecule that is produced contains the base uracil instead of thymine. So there are no T's in the base pair sequence of RNA. Whenever the RNA polymerase detects the base adenine in the template strand, it adds uracil in the forming RNA strand. So this is how the enzyme RNA polymerase adds new base pairs in the 3' end of the growing RNA chain, and this is known as elongation. The next step is termination. So just like we had a sequence of gene known as the promoter region which signaled the enzyme where to start, we have regions of the gene that basically signal the enzyme where to stop. These signals are known as terminators, and they signal that the RNA transcript is now complete. Once the RNA polymerase reads and transcribes the terminator regions, the newly formed RNA transcript is released from the transcription complex. So this was the first section of this process in which the DNA is used to produce a RNA molecule. This RNA is known as the pre-mRNA or the pre-messenger RNA. The prefix is used here to show that this mRNA is not yet ready to leave the nucleus and start the process of translation. So the next section of this process of transcription deals with how this pre-mRNA is converted to a mature RNA molecule. Imagine that you want to publish a new book and you have printed all the pages. Now you cannot sell the book yet because the new book needs covers, both front and back. Also, before selling it, it is important to check for any blank or wrong pages that may have been inserted. These two processes essentially represent how the pre-mRNA is processed. The first step involves addition of a cap and a tail to the ends of this pre-messenger RNA molecule. These caps and tails are short amino acid sequences that protect the pre-mRNA molecule. The free ends of the pre-mRNA are liable to attack from many enzymes. The cap and the tail covers these ends. The second step of this process involves removal of any unuseful or junk sequences that do not code for proteins, and these are known as introns. This is known as RNA splicing, in which the protein RNA complex known as the spliceosome removes these non-coding parts. The rest of the DNA is then joined again together to create a mature RNA molecule. So this was a brief look at the process of transcription.
So this video explains how transcription basically happens. Like I explained, there are these steps. First, the RNA polymerase comes and attaches, and then it forms the mRNA, and then this mRNA is goes through some modifications before it exits the nucleus. And one of these modifications is attaching of the cap and the tail, and then cutting out of the introns, and then it leaves the nucleus for translation in the cytoplasm. So, talking about translation, sorry for this, one second. Yeah. Talking about translation, so what are the players involved in translation now? Now that we have discussed transcription, we have to move on to the next part, which is translation, which is the formation of the protein from the mRNA. So in this, the players involved are basically the mRNA because that's the template. Then we have the ribosome, we have the tRNA, and we have the amino acids. These amino acids are the building blocks of the protein. So here is a ribosome. There is this larger subunit of the ribosome and the smaller subunit of the ribosome, and it has these three sites: A, P, and E. The A is the amino acyl site, P is the peptidyl site, and E is the exit site. And in the lower subunit is actually the mRNA binding site where the mRNA will come and bind. And to these sites, the tRNA will come and bind. And at the A site, the first tRNA binds, and then it moves on to the P site. And once this the amino acids present on the tRNA have attached to each other, then they exit from the ribosome, and the ribosome moves along the mRNA. And this is what a tRNA basic molecule looks like. It looks like a clover leaf. It has these three loops. One is called the T loop, one is called the D loop, and there is this one variable loop. And in the bottom is the important part, which is the anticodon. So basically you have these codons, or the mRNA is read as a sequence of three codons each, and three of these codons, they code for one amino acid. So this anticodon fits to these codons on the mRNA, and complementary to these, the amino acids are attached on the 3' of the tRNA, and these amino acids are complementary to the codon present on the mRNA. So they are then attached to each other. So this is how the tRNA transfers information from the mRNA, the messenger RNA, to the amino acid sequence or the polypeptide chain. So how transcription happens is, now as I mentioned, this is the mRNA, and on the mRNA, the tRNAs come and attach. So you have the start codon AUG. I will come to codons in a while. So you do have this sequence, and three of these base pairs, they are called one codon, and there are no gaps in these base pairs. The consequent ones are the next base pairs. So these three base pairs are attached to, this is the anticodon UAC. So if the codon is AUG, so against A will come K against A, and then against G will come C. So this is the anticodon, and for this codon AUG, methionine is the amino acid that is attached. Then to the next one, and the messenger RNA is being read from 5' end to 3' end. To the next one attaches this next one, which is valine, and to CCC attaches glycine, and then it forms the protein altogether. So if you look at these different steps, 1, 2, 3, 4, in the first one, at the A site comes and attaches one tRNA, and then the A site shifts to the P site, and then another one comes to the A site, and then these two gets bound together, and then that moves on to the E site and exits, and then next one comes to the A site. So it goes on like a cycle, and it makes the long polypeptide chain. Again, you can see here, first one, a new tRNA binds the lattice on the A site, then the tRNA leaves the E site, and the next, the ones on the P moves to the E site, and the one on the A moves to the P site, and like that it keeps moving forward to form the entire polypeptide chain. Okay, so this is another short video just to explain translation a little better.
So the process of translation occurs in three basic steps. First step is known as initiation, in which the ribosome assembles around the target mRNA. In the second step, known as elongation, the tRNA transfers amino acids to the ribosome, which are joined together to form a polypeptide chain. In the last step, known as termination, the ribosomes release the polypeptide when it reads a stop signal on the mRNA.
Okay, so coming to the codons that I was talking about earlier, so basically every three nucleotide bases in the DNA sequence, they code for a, so three of them code for a specific amino acid. So you can see in this table, if in the first position you can have either U, C, A, or G, in the second position again you can have U, C, A, or G, third position again the same, and then the different combinations of these give rise to different amino acids. So if you have UUU, it's phenylalanine. If you have UUA, it's leucine. If you have UCU, it's serine. So some of these are marked here. So the one in green is methionine, which is always the start codon. So to keep in mind, AUG is the start codon, which always leads to the starting of the polypeptide chain. And there are three stop codons, which lead to the, which is the last codon in the polypeptide. And after that, so basically, after that, no amino acid is attached. So UAA, UAG, or UGA, these three codons, they mark the end of the polypeptide chain, or they tell the tRNA to stop, and no further amino acids are attached. One thing to keep in mind is that you can see that a lot of these, there is a redundancy. So we only have 20 amino acids, but if you look at this combination of these four pairs and the three and the in sequences of three, when they are coding for amino acids, there are actually 64 combinations, but they only code for 20 genes. And if you take out these three stop codons, then there are still 61 combinations, but they're coding for only 20 genes. So there is a redundancy in the sense that multiple codons are coding for the same amino acid. You can see here in this section, all of them are coding for threonine. Here, AUU, AUC, AUA, all of them are coding for leucine. All of these four are coding for valine. So you can see that there is some sort of redundancy. And one thing to keep in mind is that this last base pair, is called the wobble base or the wobble position, because in a lot of these places you can actually replace the last base pair with any one of the with any other one, and it will still be coding for the same amino acid. So for example, in glycine and arginine, you can replace the last pair with any of the four combinations. It will still code for the same amino acid. So that's why it's called the wobble position. Okay. This is what you can see here. This is the wobble position. If you have the first two correct, then the last one can be anything. It will still code for the same amino acid. For example, in the case of leucine, proline, arginine, or glycine, most of these, alanine, valine. So this is the wobble position. So, coming to the types of RNA polymerases in eukaryotes, as I mentioned when talking about the difference between eukaryotes and prokaryotes in terms of their uh, transcription. So, in transcription, prokaryotes only have one kind of RNA polymerase, which functions for transcribing everything, proteins and RNAs and everything. But in eukaryotes, we have three different types of RNA polymerases. Each type transcribes different types of genes. So, RNA polymerase 1 is actually the one that transcribes the largest ribosomal RNA. So, ribosomal RNA is one of the um, it's the RNA that forms a part of the ribosome complex. So the RNA polymerase 1 transcribes the three largest ribosomal RNA subunits, which is 28S, 18S, and 5.8S. And this is located in the nucleus. RNA polymerase 2, which is the one that is most frequently talked about because it transcribes protein coding genes, and it transcribes mostly mRNA, microRNA, snRNA, which is small nuclear RNA, and snORNA, small nuclear RNA. And it requires transcription factors to bind to the promoters. So, transcription factors is another term. So, these are more proteins which actually help binding the RNA polymerase to the promoters. Promoter is the sequence that I mentioned, which is present in front of the gene, which actually marks the starting of the gene and where the RNA polymerase comes and attaches. So, transcription factors enhance the attachment of the RNA polymerase to these promoters to increase the transcription rate. And the third RNA polymerase, it transcribes tRNAs or transfer RNAs and the smallest rRNA subunits, which is the 5S rRNA. tRNA, as you now by now know, it's very important in translation, and this is what translates the information from the anticodon to the amino acid. And it also transcribes other small RNAs which are involved in splicing and protein transport, such as small nuclear RNA and scRNAs. Okay. Moving on to the different types of RNAs. So now that we've discussed the different types of RNA polymerases, and we've discussed how many types of different types of RNAs they produce, there are different types of RNA. For example, there is mRNA, which is the basic one, which is the messenger RNA, which then encodes for proteins. There is tRNA, which is transfer RNA. It acts as the adapter between the mRNA and the amino acid. So it transfers the information from the mRNA to the polypeptide chain. rRNA forms the ribosome. So, as we talked about the large subunit, which, as discussed, has these 28S, 16S, 5.8S, and the small one has the 5S. Then, small nuclear RNA, these they functions in various processes. For example, splicing. Then, small nucleolar RNA. So, inside the nucleus, you might be aware that there is this place called the nucleolus, which is also present. So, it's present inside the nucleolus, and it facilitates chemical modifications of the RNAs. Then microRNA or miRNA, it actually regulates gene expression. It can play a very important role in gene regulation, which we'll talk about later. Small interfering RNA, also called siRNA or RNAi, these are again they play an important role in gene regulation, and these are actually exogenous. They're not generated inside the cell. MicroRNAs are actually endogenous, and they are formed or created inside the cell, but siRNAs are not created inside the cell. And there is this long nuclear RNA, lncRNA, which also regulates gene expression and also regulates the activities of miRNA and siRNA. So, moving on to some inhibitors of protein or RNA synthesis. Now that we have so much of protein being synthesized in the cell, so much of RNA being synthesized, we there might also be something to have a check on it. There might also be something that inhibits, because in nature, not everything cannot go on and on. There is always a check on something that is happening. So there is inhibition of protein and RNA synthesis as well. So an inhibitor of protein or RNA synthesis. So there is an inhibitor which only acts on bacteria. There are two of these examples of antibiotics. There is tetracycline and rifamycin. How tetracycline works is that it blocks the binding of amino acyl tRNA to the A site of ribosome. So, as we discussed, the A site of ribosome or the amino acyl site of ribosome, it blocks the binding of tRNA there only. So, again, so of course, translation cannot happen. And rifamycin, it actually blocks the initiation of RNA chains by binding to the RNA polymerase. So, it prevents the formation of the mRNA itself. But these two antibiotics only work on bacteria. This next one acts on bacteria and eukaryotic cells as well. It's actinomycin D. It binds to the DNA and blocks the movement of RNA polymerase. So if RNA polymerase cannot move along the DNA, then it again prevents the RNA synthesis. So mRNA cannot be formed. And next one, which acts on eukaryotes but does not act on bacteria, one is cycloheximide, which blocks the translocation reaction on ribosomes. So it blocks the reaction of translocation in ribosomes, which means the movement of tRNA from A site to P site. And alpha amanitin, which blocks mRNA synthesis by binding preferentially to RNA polymerase 2. So it preferentially, so this amanitin, it binds to RNA polymerase 2, and therefore it does not allow RNA polymerase to find mRNA. Okay. So moving on to in the end of this week, they provided a revised version of Central Dogma based on what all we have learned. So DNA does form RNA by transcription, and RNA can form different types of proteins by translation. The same RNA can form different types of proteins based on which exons are present in this RNA. So as we discussed, that there are exons and there are introns. So when you're splicing, you can actually jumble up the position of the exons. You can put exons in different areas. For example, there was a sequence of A, B, and C. You can actually have different exons connected. You can connect them as BCA or BAC, and these will give rise to different polypeptide chains. So it this is another level of redundancy. So you can form different proteins from the same RNA also by uh, translation. And then protein can give by alternative splicing and by translation. As I discussed, RNA can give rise to different proteins, and RNA can also give rise to other nuclear RNA or microRNAs. Okay, RNA by reverse transcription can give rise to RNA by reverse transcription. It can also give rise to DNA, which is an RNA can give rise to other RNAs, which happens in a lot of viruses. Okay, moving on. So, talking about what is gene regulation and why do we actually need it? Uh, So, let's discuss what is gene regulation and why is it required. So, genes cannot always stay on, right? So, as I discussed, everything is mostly regulated in nature. So, some genes are required to always be on. For example, the genes that play a role in the beating of our heart, in controlling how we are breathing, in all these essential things that have to go on um, throughout our life, then those are the genes that always need to be on. For example, proteins that help in the beating of our heart or in these things. So that's called constitutive expression. But other genes are only required when there is certain change in environment. For example, when you've consumed food, only then do you require genes that produce enzymes that are required in our digestive pathway. So these genes are regulated in some sort of a way. They are switched on only when they are required by the body. And again, during the development, say from an embryo to a full grown adult, there is a lot of the times when the cells get differentiated. So you might already know that when an embryo is formed, when a zygote is formed by the fertilization of the egg by the sperm, then it forms a blastocyst which has a lot of these cells, and some of these get differentiated into forming different organ systems. For example, the circulatory system, nervous system, excretory system, digestive system. So all of these cells, they actually possess the same DNA. They have the entire gene sequence, they have the entire instructions, but they prefer or they choose to you follow only some of these instructions, and that is how differentiation happens. So the cells in the circulatory system will follow only the instructions that are required for them to function in the circulatory system, and they will not follow the instructions that can actually help them to become glands or can help them to act as digestive organs or things like that. And the cells that form a part of digestive tissues, they also have the capability or the ability. They also have the same instruction sets as the other. They have the same DNA, but they also only follow the instructions that help them into fulfilling their role in the part of the digestive tissue. Okay, so that is how during development gene regulation plays a very important part in cell differentiation. So, gene regulation is again different in prokaryotes versus eukaryotes. In prokaryotes, genes are usually on, and for regulation, they are switched off. So, in prokaryotes, most of the genes in their base state, they're on. So, they're always being transcribed and translated, they're always producing polypeptides. But for regulation, you mostly switch them off. So, when you don't require them, is when you switch them off. In eukaryotes, this is the reverse. Mostly the genes are off, but when you do require them, you turn them on. So we are not always producing, in eukaryotic cells, they're not always producing polypeptides, but only when that particular protein is required, that enzyme is required, only then the gene is turned on. Okay, I hope this is clear. Moving on to what are the different mechanisms of gene regulation in a cell. So for gene regulation, you can have DNA methylation. This is what, so DNA is present. So as I said earlier, chromatin modifications. So DNA is a double helix, and when DNA methylation happens, that is when it makes the DNA inaccessible to RNA polymerase. If a part of the DNA is heavily methylated, it's properly coiled up. Then it becomes inaccessible to the RNA polymerase. And then the RNA polymerase cannot actually access those portions of the gene. For example, so if there is a cell which forms a part of the digestive tissue, it might have other instructions that help in becoming a muscle, or that help in becoming an artery cell, in a vein cell, by blocking those areas by heavy DNA methylation of those portions, which include genes that help the cell becoming those areas, and it keeps only those genes accessible that help it in becoming a digestive tissue. So DNA methylation, it actually makes those some genes inaccessible to the RNA polymerase molecule or enzyme. Then there is differential gene transcription. Only some genes get transcribed while the others do not. So there is a differential gene transcription. Selective RNA processing, as we know, RNA is then processed. Some of the exons are kept, introns are cut out, and these exons can be jumbled around. So that is selective RNA processing. Give me a minute. My network is a little stuck. I will turn off my camera for a while because the network here is a little patchy. Yeah. Okay. So we were discussing about these different DNA regulation mechanisms. So I was saying that there is selective RNA processing. So as we discussed, the RNA, the mRNA, pre-mRNA gets processed before it becomes the mature mRNA. So during this processing, you actually cut out the introns and you keep the exons. So these exons can be jumbled up to make different types of genes or different types of polypeptide chains, and that is another form of gene regulation. Then there is selective translation. Even after a mature RNA moves out of the cytoplasm, moves out of the nucleus into the cytoplasm and is ready for being transcribed, it can still be blocked. Its translation can still be blocked, and only some mRNAs will get translated. So that is called selective translation. And we'll discuss in some time how these can still be blocked by other RNAs. And there is protein modification. The polypeptide chain that is being formed, it can further be modified, which leads to which adds another layer of gene regulation. Okay. Talking about the components for gene regulation. So in the I Think Biology book, they've given this table which discusses about the different components that play a role in gene regulation. So first of all, in the DNA, within the DNA which we discussed, it is the genetic material which is inherited and which is replicated during each cell division. So the different components in the DNA, first of all, is the gene. Gene, as we know, is that sequence or the segment of the DNA which codes for for an RNA molecule or for a polypeptide chain, and it's the basic unit of heredity. So it's the segment of DNA that basically encodes for protein. Then there is an operator sequence. This is mostly present only in prokaryotes, and this is a segment of DNA on which a repressor comes and binds. So it basically, uh, as we discussed in prokaryotes, all the sequences are on, or all the genes are always being transcribed, and you actually need a repressor to switch off the gene. That is why in prokaryotes you have this operator sequence where the repressor comes and binds and it switches off the transcription of that gene. In eukaryotes, you have a promoter segment. The promoter segment is where the RNA comes and binds. And there is an enhancer segment, which is a segment of the DNA where various modulators, for example transcription factors, as we discussed, which are another protein molecules, these transcription factors, they bind to the enhancer segment, and they in turn help to recruit the RNA polymerase to the promoter. So these transcription factors, they are proteins that come and attach to the enhancer, and then these transcription factors bring in the polymerase, and they recruit the RNA polymerase to the promoter and lead to switching on of that particular gene. Then, having discussed the DNA, comes the RNA. RNA is the thing that is produced after transcription from the DNA. It has exons and introns, as we discussed. Exons are those portions of the mRNA that are retained and they're joined together after splicing, and these are the ones that actually code for protein. So they are the ones that get translated in the end. And introns are the portions which are spliced out because they do not code for proteins. They are sometimes called junk DNA, but we now know that they are not junk because they might be coding for other things like small nuclear RNA or small interfering RNAs or microRNAs. But these do not get translated into proteins. And then there are non-coding RNAs. Non-coding RNAs are the RNAs that are not translated into protein, but they form a part of gene regulation. They play other regulatory functions. Then in the end is the protein. That is the end product of transcription and translation. So it has transcription. So the proteins that play a role in central dogma are actually transcription factors. As we discussed, transcription factors are proteins that recruit the RNA polymerase enzyme to the DNA. They're also called activators. And there is RNA polymerase, which is the central enzyme that is responsible for um, transcribing the DNA into the complementary mRNA strand. So it reads the DNA and forms the complementary mRNA strand. So these are the components that are required that play a role in gene regulation. Okay. So coming to non-coding RNAs, there are different types of non-coding RNAs. First type includes tRNA and rRNA, which is the transfer RNA and the ribosomal RNA. They're present in the cytoplasm and they help in the translation of mRNA to protein. So rRNA is the one that forms the ribosomal subunits, and to the rRNA, mRNA comes and attaches to the RNA, and the tRNA then, as we discussed, translates the information coded in the codons of the mRNA to the amino acids in the polypeptide chain. So they help in the translation of mRNA to protein. snRNA, which is also called the small nuclear RNA, as the term suggests, it's located inside the nucleus. It is about 150 micronucleotides long, so it contains about 150 nucleotide bases, and it regulates the transcription factors. Now, we've already discussed transcription factors are actually the proteins which enhance or which which promote the um, attachment of the RNA polymerase enzyme to the promoter sites of the DNA. Then there are microRNAs, miRNAs. They are single stranded RNAs. They usually end up forming hairpin loops by folding upon themselves. They are endogenous, which means that they are produced inside the cell. And they're about 20 to 24 nucleotides long. And um, they have a role in post transcriptional regulation, which we'll be discussing in the next slide. And they can block mRNA from being translated. So they can block the mRNA that has actually left the nucleus and is in the cytoplasm from being translated. siRNA, which is also called small interfering RNA, it is again 20 to 24 nucleotides long. But the major difference between si and miRNA is that this is exogenous. It is not formed or not created inside the cell itself. It comes from external sources. For example, viruses. And because it comes from external sources, you can take a guess that it forms an important, it can be nicely used in biotechnology and can be introduced by humans, can be introduced by humans to actually control the gene regulation, to control which proteins are being made in the cell. And then long nuclear RNAs, they're around 200 nucleotides long, and they regulate the transcription of both microRNAs and siRNAs, and they work in chromatin modifications, which is another way of regulating genes or gene transcription. Then, what is the mechanism for miRNA and siRNA? As I said, so these miRNAs, you can see here, this is the microRNA, which forms a hairpin loop. And these are the synthetic, or the input could be a synthetic siRNA. There is this dicer. So dicer is another enzyme, and what it does is basically it breaks off this hairpin loops and converts them into single stranded RNA molecules. So now, currently it is double stranded because the RNA is folded in upon itself. So the dicer breaks this fold apart and converts it into a single strand, and this single strand then connects to this RISC complex. And this RISC complex, along with the single stranded miRNA or siRNA, it goes and attaches to the target mRNA, and it then prevents the transcription of this mRNA, or it leads to the degradation of the target mRNA. Okay, so this is how microRNAs and interfering RNAs, they prevent the transcription of a mature mRNA into a protein. Okay. So, how was this microRNA discovered? So, it's a very interesting story. It's a very interesting anecdote. Basically, there was these two researchers, Victor Ambrose and Gary Ruvkun. They got a Nobel Prize for this discovery. They were studying C. elegans, Cenorhabditis elegans, which is a nematode. And C. elegans has these different stages of larvae. They have L1 larvae, L2 larvae, L3, and L4 larvae during their development. So they were studying these C. elegans and different mutant form of these elegans. So while studying C. elegans, they came across a mutant form which could not uh, go ahead from the L1 stage. So it was stuck in the L1 stage, could not move on to the L2 stage. So this is when they actually discovered a tiny RNA molecule which is just 22 nucleotides long. That is why they named it microRNA. And this, they found that it's they called it lin4. So this lin4 microRNA was actually preventing the activity of the mRNA of lin14. So the gene is called lin14, and this lin14 gene is required for the larvae to move ahead from the L1 stage to the L2 stage. So but this gene was not being transcribed in this L1 larvae, in this mutant L1 larvae which had this lin4 microRNA. So this lin4 microRNA is actually acting upon the mRNA of lin14 and preventing it from making this protein, which prevents it from moving forward from L1 to L2. So you can see here the microRNA, the miRNA lin4. So this here lin4 is actually a microRNA, and this microRNA lin4, when its amount rises, when its levels rise, that is when the levels of lin14 come down. So the mRNA lin14 comes down, and this gene does not get or this protein does not get made, and that is why from L1 to L2 it's unable to yeah, that is why it's unable to progress from L1 to L2. And then later they also found that from L4 to reaching to adult, there is this lin41 which is required. Lin41 is essential for converting from L4 to adult. But this was not happening, and the larva was stuck in the L4 stage. That was because of the presence of another miRNA, which is called LET7. And this LET7 was inhibiting the formation of Lin41. So, this is how these microRNAs were actually discovered in C. elegans. And for this discovery, Ambrose and Ruvkun got a Nobel Prize. And that is how we got to know about microRNA. Okay. So this was all that was covered during this week, and now we will be moving on to the question answer session of today, where I will be giving you some basic questions based on this week's information, and these questions might help you in your in answering this in attempting your exam, and will prepare you for your exam. Okay. So the first question is, what is the correct sequence of information flow in the central dogma of molecular biology? So we discussed central dogma. So the question asks, what is the correct sequence of information flow in central dogma? Is it from protein to RNA to DNA, from DNA to RNA to protein, from RNA to DNA to protein, or from DNA to protein to RNA? So what is the correct one? So I will give you 5 seconds to guess the answer. So the answer to this question is actually DNA to RNA to protein. As we've already discussed, DNA is the basic genetic instruction which is present or which is shared by all of the living organisms. And from DNA, after transcription, we get RNA. And from this mRNA, it's then transcribed with the help of tRNAs and rRNAs into the protein sequence. So that is what is the basis of central dogma, as was proposed by Crick, Francis Crick. Next question. Which enzyme is responsible for converting DNA to RNA during transcription? So, what is the enzyme that helps in the forming RNA from DNA? Is it DNA polymerase, RNA polymerase, reverse transcriptase, or ligase? So the correct answer to this question is RNA polymerase, as you might already be aware. So what DNA polymerase does is DNA polymerase is required for DNA duplication. So when you are during cell division, when a DNA is making its own copy, that is when DNA polymerase plays a role. So it uses the DNA, both the strands of the DNA, as template strands and makes the uh, complementary strand to that. So that is DNA polymerase. RNA polymerase, on the other hand, uses the DNA template strand in order to make the mRNA, which is then used for making proteins. Reverse transcriptase, as we discussed, is the enzyme that actually is present in some viruses, which leads to RNA forming DNA back. So that is reverse transcription. And ligase are enzyme that joins together the different nucleotide sequences. For example, when exons need to be joined together, that happens by it actually joins the Okazaki fragments together, which you will learn about more when talking about DNA duplication. Coming to what type of RNA carries amino acids to the ribosome. So, what is the type of RNA which is carrying amino acid to ribosome? Is it mRNA, rRNA, tRNA, or snRNA? I'll give you 5 seconds to think about the answer. So, the correct answer to this question is tRNA, also called the transfer RNA, as we already discussed. The tRNA or the transfer RNA has three different loops, and the bottommost loop is actually the one that has the anticodon, which attaches to the mRNA on the codon. And on its 3' end, the tRNA has the amino acid complementary to the codons. So that the tRNA along with the amino acid, it brings it to the ribosome, and the ribosome on the A side, it comes and attaches. And so that is how it brings the amino acid to the ribosome. mRNA is actually the messenger RNA, which is formed by the transcription of DNA. rRNA forms the subunits of ribosome, and snRNA is the small nuclear RNA. Okay, next question. Where does transcription occur in eukaryotic cell? So, what is the location where transcription occurs in eukaryotic cells? Is it the cytoplasm, nucleus, mitochondria, or the ribosome? So, the correct answer to this is the nucleus, as you might already be aware. As we discussed, eukaryotic cells have compartmentalization and transcription, which is the formation of mRNA from DNA that actually happens inside the nucleus. What is the product of transcription? So, what is the outcome of transcription? Is it DNA? Is it RNA, protein, or amino acids? What is the product of transcription? So, the answer to this question is RNA, as we have discussed at length during this tutorial. Transcription is actually when RNA polymerase, the enzyme RNA polymerase, it reads the DNA template strand and forms the complementary RNA strand. So that is the product of transcription, mRNA, to be more precise. Next question asks, what is the function of a promoter in transcription? So, what function does a promoter play in transcription? Option A, it binds RNA polymerase to initiate transcription. Option B, it terminates transcription. Option C, it splices out introns. Or option D, it translates mRNA into protein. So, what is the function of promoter in transcription? So, the correct answer to this question is actually option A, where it binds the RNA polymerase to initiate transcription. As we already discussed, promoter sequence is a sequence which is present right before the gene. And this is where the RNA polymerase comes and binds, and then it starts transcribing the gene. Next question. In eukaryotes, what happens to pre mRNA before it leaves the nucleus? So, what happens to pre mRNA before it's leaving the nucleus? Is it immediately translated? It is degraded by the nucleases. It undergoes splicing, capping, and polyadenylation, or it is converted into DNA. So, what happens to the pre-RNA molecule when before it's leaving the nucleus? Yeah. So, the answer to this question is that it undergoes splicing, capping, and polyadenylation. Okay. So, I will set a timer for 5 seconds for the upcoming questions so that, or for 10 seconds so that you guys can take that much time to think about the answer. So, as we discussed, before leaving the nucleus, the pre mRNA gets processed and it mostly undergoes splicing where the exons are kept and the introns are removed. It goes capping at the 5' and the polyadenylation or the poly A tail, where adenine, the amino acid adenine, is attached, and at the 3' and it undergoes polyadenylation. Fine. Moving on to the next question. What is the start codon for translation? So, what is the start codon for translation to become? Is it UAA, AUG, UGA, or UAG? So, I'm starting the timer. Yeah. So, the answer to this question is AUG. So, AUG, as we discussed, codes for methionine and it's the start codon for translation. Moving on to the next question. Next question is, the bond formed between amino acids in a growing protein chain is called what? So, what is the bond that is formed between amino acids in a growing protein chain? Is it a hydrogen bond? Is it a peptide bond? Is it an ionic bond? Or is it a phosphodiester bond? So, that was the 10 seconds timer. So, the answer to this question is actually the peptide bond. So, as we know, the amino acids come together and form a polypeptide chain. So, from there, you can guess the answer that it's a peptide bond mostly. Okay. So, Question number 10. What is the role of codons in translation? So, what roles do codons play in translation? Do they signal where transcription should start? Are they sequences of three nucleotides that code for amino acids? They help fold proteins, or they act as a blueprint for DNA synthesis. Your 10 seconds timer starts now. Yeah. So the answer to this question is that codons are actually the sequences of three nucleotides that code for amino acids, as have been discussed in this tutorial, as well as you might already know from high school. Codons are basically sequences of three nucleotides, and they code for amino acids. And keep in mind that there is no overlapping or there is no gaps in between codons. One codon is just followed by another codon. Okay. Next question. Which of the following biomolecules is not a macromolecule? So, which of the following biomolecules is not a macromolecule? Carbohydrates, proteins, lipids, or vitamins? So, the answer to this question is actually vitamins. You might already be aware, or we discussed. That carbohydrates are actually macromolecules formed from simple sugars, proteins are formed from amino acids, lipids are formed from fatty acids. So, they're all macromolecules, but vitamins, they are not. Next question. What is the monomer of lipids? So, what is the monomer of lipids? I already gave away the answer in the last question. Is it nucleotides, amino acids, fatty acids, or monosaccharides? So, the answer to this question is fatty acids. Lipids are formed from fatty acids. What is the role of transcription factors? So, what role do transcription factors play? Do they break down mRNA, controlling the expression of specific genes, transporting of amino acids, or synthesizing lipids? What is the role of transcription factors? So, the correct answer here is controlling the expression of specific genes. So, as we discussed, transcription factors are proteins which actually attach to the enhancer segments, and they then recruit the RNA polymerase to the promoter segments of the DNA, and that is how the um, transcription begins. So, they control the expression of specific genes. Moving on to the next question, question 14. In prokaryotes, gene regulation commonly occurs at the level of what? At the level of transcription, translation, DNA replication, or protein folding. The time starts. So, the answer is transcription. In prokaryotes, gene regulation is pretty basic and it only happens at the level of transcription. Question 15. What is the main purpose of gene regulation? So, what is the purpose of gene regulation? Why do you need it? So, is it to allow all genes to be expressed at all times, to control when and how much a gene is expressed, to prevent the formation of proteins, or to remove non coding DNA? So, the correct answer to this question is to control when and how much a gene is expressed. So, as we have discussed, gene regulation helps you control when and how a gene is expressed. So, in prokaryotes, a gene is always on, and for regulation, it is switched off. And in eukaryotes, a gene is always off, and for regulation, it is switched on whenever required. 16th question. The difference between microRNA and siRNA is that microRNA and siRNA are both exogenous. MicroRNA is endogenous, but siRNA is exogenous. MicroRNA and siRNA are both endogenous, or microRNA is exogenous and siRNA is endogenous. So the answer to this question is that microRNA is endogenous and siRNA is exogenous, as I have already discussed during the tutorial. Last question for today. Choose the only group with non-coding RNAs from the following groups. Choose the group which only has non-coding RNAs from the following group: tRNA, rRNA, snRNA, miRNA, rRNA, siRNA, lncRNA, miRNA, snRNA, miRNA, lncRNA, miRNA, or tRNA, siRNA, and miRNA. So, which of these has only non-coding RNAs? So, actually, the answer to this question would be all the four options, because none of these options um, has mRNA, which is a non. So, all of these are actually sorry. The correct answer to this question is the third option, which is small nuclear RNA, micro RNA, LNC RNA, and micro RNA again, because tRNA and rRNA are involved in coding. So that was all for today, and thank you for watching this video. And my name is Geetika Agarwal. I am a PhD student at the Center for Ecological Sciences in IISc Bangalore. If you have any doubts, you can ask in the comment section, or you can ask in the discussion forum of the NPTEL course. Thank you.